Hello everyone, welcome you back on my YouTube channel Ravishar Biology class, and today's session will be taken by me in the subject for biology for 10th standard. Some videos already uploaded to the channel as well for the 10th standard, and for 12th standard it's still going on. In this video we will discuss about nutrition in animals. Nutrition in plant, it was done in my previous lecture, and today's lecture will be done on nutrition in animals as well as nutrition in amoeba. So let us start the topic today, nutrition in animals. So these are the points, ingestion, digestion, absorption, assimilation and egestion. These topics we have already I have already introduced you, but on the board it is mentioned, but I'm going to tell you because these all 5 topics are very important for the students, those who are going to participate in 10th boards. All the animals, whether we are talking about single cell animal, the most simplest animal on the earth, amoeba, single celled animal, or any complex animal, they both kind of varieties of animals, they go through nutrition with along for all these 5 steps. All the 5 steps of these what I have mentioned on the board, they all go through these 5 steps. Let me clear once again. All these 5 steps are common in unicellular animal as well as multicellular animal. So if we talk about amoeba, the single cell organism, or if we talk about the human being, we all just go through all these 5 steps. So here we have to discuss about these 5 steps, and afterward we will discuss about how these 5 steps will be applied in amoeba. So let's start. The first one is ingestion. Ingestion means intake of food. How an animal taking food inside the body is called ingestion. After that, the food material you have taken is to be digested into the body, means the complex food material will be broken down into the simpler part, and this is called digestion. Now the digested part is to be dissolved into the blood, so that blood will transport each and every cells of our body. So this will go into our blood by the villi, and so many structures are there which we will discuss in nutrition in human. So absorption will be done into the blood. This process is called absorption. The fourth topic is assimilation. Assimilation means the food has been distributed to each and every cell after absorption and digestion. And the last topic is egestion. The food material has been digested, has been absorbed, assimilation was done, but some of the food material is remain left as it is not get digested into the body, into the alimentary canal or into the organism's body, and therefore it has to be removed out of the body, as they are not very good for the body. They will be toxic for the body, harmful for the body. Therefore the material which is not digested, the food material which is not digested, has to be removed out of the body, and the removal of these wastes are called, the process is called egestion. So this is all 5 steps which is very common in all the organisms, whether we are talking about single cell animal or the multicellular animal. Today we will talk about one of the most important part of this segment, nutrition in an animal, is nutrition in amoeba. It's really very important part. So many times in the board examination it has been asked, what is the mode of nutrition in amoeba? Let me clear. The mode of nutrition is heterotrophic, as they depend for their food on other organism, as well as a type of heterotrophic nutrition is called holozoic nutrition, where a complex food completely intake by the animal, digestion will be done, and the energy will be produced by the further activities. So this is the animal. Amoeba, like we human being, they perform heterotrophic nutrition as well as they go through holozoic nutrition, a type of heterotrophic nutrition. So they perform holozoic nutrition. Let me write in a bracket holozoic nutrition. This is the variety of nutrition of heterotrophic. Nutrition in amoeba, how do they perform? So let me clear once again, as I had told you in the very beginning, that all the amoeba or complex animals like human being, dogs, cat, rat, they all go through these all 5 steps. So with the help of diagram, we can understand. Suppose this is an animal. This is the nucleus of animal. This is a single cell animal, of course, so this will be having a nucleus as well. So this is an animal, and suppose the food material, diatoms or small algae are there. This is a food material for this. So how it will take this organism? इसको थोड़ा सा नीचे करते हैं. This is the organism which is considered as the food of this animal. So it has to move towards it by forming a structure over its body. It makes a structure towards the animal so that it can move to the organism to have their food, and this structure is called pseudopodia. The singular word for pseudopodia is pseudopodium. So these are two structures, so this will be called pseudopodia. The word pseudopodia is taken from two words, pseudo plus podia. Pseudo means, this is, I'm writing here, pseudo means false, and podia means feet. The organ, not actually feet, false feet is called pseudopodia. So pseudopodia will be produced towards the food, and this animal amoeba will cover the organism properly, as it can change its shape according to the need. It has been changed here now, from here to here it has been changed. Some shape changes will be found here, and it is now covered with the organism. Still it is not taken in. So what will be happen? The breakdown of the membrane will be taken from here as well, and it will be now intake by the amoeba, and this complete process is called ingestion, as the food has been taken inside the cell of amoeba. So this is how an amoeba is taken its food. Now it's time to go through the second step, and this is digestion. Means the breakdown of food will be taken, as they don't have teeth, they do not have the alimentary canal like we people, as they are having only one cell. So this organism have some specific feature. Let me clear that specific feature. Suppose you have taken a plate like thali, and you placed a bowl full of some food material, and you placed it into the, means a plate, and into which a boundary is there in that plate, which is called thali, and a bowl we have taken into which some food material is there, and we placed into the thali. We shake it, that thali, we shake it. The way of we are shaking the thali, the food material present in bowl will comes out. The movement will be done outside, and most of the food particles, food material which is present into the bowl will move out of the bowl, and it will be spreaded into the thali. Means I have considered as the food vacuole which is made. This vacuole is called food vacuole. This food vacuole will be considered as the bowl which is having food, and the cytoplasm present here, this is cytoplasm. This will be considered as the cytoplasm will be considered as the complete thali, and this is about the common example, so that you can understand the process of digestion. So this will be how it is shaking, and by the shaking, and the food vacuole will strike on this wall, here, everywhere, so that the second step will comes out automatically. How it will comes out? This is the nucleus. This is a food material, and this is connected into a food vacuole. So this will striking on the boundary or the plasma membrane of amoeba, so that the food material will move out and broken down into simpler substances. This process is called digestion. So this is how an organism is, this amoeba is performing digestion. After digestion, the food material which is digested, it has to be absorbed by the cytoplasm, and the next step will comes up, and the step will be, this will be the food vacuole. Now it has been digested almost, and this will be nucleus of this, right? So this is absorbed throughout the cytoplasm, and therefore the size is getting reduced a bit, and this is the process which we call absorption. This is the third step, absorption. In the next step, the food material which is absorbed into the cytoplasm, it has to assimilate into the cytoplasm. Assimilation means proper distribution into the cell. After absorption, the distribution will be done so that the energy production will be possible. So in the next step, the food vacuole will be looking small in size because of absorption is done, and it has been spread. The food, digested food, is absorbed by the cytoplasm. Somewhere in the cytoplasm, the nucleus is also present. So this is the process which we call assimilation. Assimilation. And the last step of the process is this food material which is still in the food vacuole, as it is not absorbed because this is not digested properly. So it has to be digested, and the digestion is not done. It means it is not a part of food material which is digested by the animal. It means very clear, the thing is, this is not the material which can be digested by the cytoplasm of amoeba, and therefore it has to be removed out of the body. And how is it possible? This food material, this food vacuole, will go to one end or near to the membrane of the amoeba. So in the next step, this vacuole will go to a place where it can be easily removed out, as it is undigested food and not good, not healthy for the animal. It becomes toxic for the animal, so it has to be removed from here. So what will be done by this animal? It will take this food vacuole near to the plasma membrane. Broken down of plasma membrane will be taking place, and this animal is now dissolved its plasma membrane according to the need, and this food material which is not digested for the animal will moves outside, and it is gone, exit, it is going through exit, and this process is called egestion. This complete process is called egestion. So these are the 5 steps of nutrition. Here we discussed completely about the nutrition in amoeba. 2-mark question boards has been asked so many times from the same segment. So let me explain the thing what I have taught today in Hindi, so that the concept will be more clear to the one who is not able to understand the English very properly. आज का सेगमेंट था न्यूट्रिशन इन एनिमल, और न्यूट्रिशन इन एनिमल में मैंने बताया कि कोई भी एनिमल हो, चाहे वो कॉम्प्लेक्स एनिमल हो या कोई सिंपल एनिमल हो अमीबा की तरह सिंगल सेल एनिमल, वो सभी के सभी 5 स्टेप से अपना न्यूट्रिशन कंप्लीट करते हैं। और वो स्टेप्स हैं, सबसे पहला स्टेप है इंजेशन, फिर उसका ब्रेक डाउन, डाइजेशन, फिर ब्रेक डाउन जो हुआ उसके बाद उस फूड को एब्जॉर्ब करना, दिस इज कॉल्ड एब्जॉर्प्शन। 4th स्टेप है कि जो एब्जॉर्ब हो गया उसको पूरे सेल्स में फैलाना जिससे एनर्जी मिल सके, एसिमिलेशन। और कुछ ऐसे फूड पार्टिकल्स हैं जो डाइजेस्टेड नहीं हुए तो उसको बॉडी से बाहर निकालना, इजेशन। तो ये 5 स्टेप्स हैं, चाहे वो अमीबा हो, डॉग हो, एलिफेंट हो या ह्यूमन बीइंग हो, ये सभी के सभी ये 5 स्टेप से ही होके गुजरते हैं। तो ये 5 स्टेप्स वही हैं जिसकी बात मैं अभी आपसे कर रहा था। तो वो 5 स्टेप्स अमीबा में भी होंगे। जैसा कि मैंने बताया कि कोई भी एनिमल हो, चाहे अमीबा हो, सबसे सिंपल एनिमल एक सेल का बना हुआ सिंगल सेल एनिमल, या मल्टी सेलुलर एनिमल ह्यूमन बीइंग हो, सभी ये 5 स्टेप से ही अपना न्यूट्रिशन कंप्लीट करते हैं। कैसे करते हैं देख लेते हैं। यहाँ पे इस डायग्राम में ये अमीबा है और रेड कलर का मैंने फूड दिखाया है। इस फूड की तरफ ये मूव करना चाह रहा है और वो मूवमेंट कैसे होगा, अमीबा मूवमेंट होता है स्यूडोपोडिया से, तो स्यूडोपोडिया हेल्प करेगा इसको यहाँ पे मूव करने के लिए। स्यूडोपोडिया 2 वर्ड से बना हुआ वर्ड है। स्यूडो मीन्स होता है फॉल्स, बायो में स्यूडो वर्ड जहां कहीं आए कंसीडर करेंगे स्यूडो मीन्स फॉल्स। तो ये फॉल्स एंड पोडिया मीन्स फीट, पैर की तरह काम तो कर रहा है, यहां से यहां आने का काम कर रहा है, ये तो पैर ही है, बट एक्चुअली ये सिंगल सेल एनिमल है तो पैर हो नहीं सकता। देयरफॉर दिस इज कॉल्ड स्यूडो प्लस पोडिया। स्यूडो मीन्स फॉल्स, पोडिया मीन्स लेग। ऐसा पैर जो फॉल्स टाइप का हो तो वो यहां आएगा। फूड कवर हो गया, यहां से ब्रेक डाउन हुआ और यहां से इजिली ये क्या हो गया, बॉडी के अंदर, ये बॉडी के अंदर कंप्लीटली आ गया। फूड वैक्यूल इसके साइटोप्लाज्म में चला गया। तो ये प्रोसेस हो गया इनटेक ऑफ फूड। खाने को बॉडी के अंदर लेना ही इनटेक ऑफ फूड है, जिसको इंजेक्शन कहते हैं। ये प्रोसेस सबसे पहले फूड को अंदर ले लिया बॉडी के, ये इंजेक्शन हो गया। फिर दूसरे में जो फूड है उसका ब्रेक डाउन होगा। वो कैसे होगा, देखो एक थाली लेते हैं, थाली कंसीडर करते हैं पूरा अमीबा, और उसके अंदर ये फूड वैक्यूल बन गया, जो फूड इसने खाया एक वैक्यूल बन गया ये है। और उसके अंदर बाउल ले लेते हैं, एक थाली लिया जिसपे बाउंड्री हो और एक बाउल लिया, बाउल में फूड मटेरियल डाल लिया और उसको हमने उस थाली में रख लिया और ऐसे ऐसे शेक करेंगे चारों तरफ। जब शेक करेंगे तो ये जो शेकिंग हो रहा है, इस शेकिंग की वजह से फूड का जो बाउल है वो कभी इस दीवार पे लड़ रहा है, इधर दीवार पे, दीवारों पे लड़ने की वजह से उसके अंदर जो फूड है वो बाहर आने लगेगा। तो ये क्या हो गया, ब्रेक डाउन हो गया, और वो ब्रेक डाउन हो गया मतलब डाइजेशन हो गया, और जैसे बाहर आएगा उसको सेल जो है वो साइटोप्लाज्म एब्जॉर्ब कर लेगा। तो ये प्रोसेस हो गया एब्जॉर्प्शन। पूरी तरह से पूरे साइटोप्लाज्म में ये फूड फैल गया। रेडिश कलर में मैंने इसीलिए दिखाया है डॉट डॉट्स में, शेड्स में। ये दिखा रहा है कि फूड जो है जो एब्जॉर्ब हुआ है वो पूरे साइटोप्लाज्म में फैल गया, उसे एनर्जी मिल सकती है। कुछ ऐसे फूड पार्टिकल्स हैं इसी में जो डाइजेस्ट नहीं हो रहे हैं, तो वो हार्मफुल हो सकते हैं, टॉक्सिक हो सकते हैं, इसीलिए इसको बाहर करने का यह प्रोसीजर है। सो दिस रिमूवल ऑफ फूड मटेरियल, यही फूड मटेरियल को बाहर करना बॉडी से इजेशन होता है। तो इस तरह ये 5 स्टेप्स हो गए। सो दिस इज हाउ वी हैव स्टडीड अबाउट न्यूट्रिशन इन एनिमल एंड न्यूट्रिशन इन अमीबा। इन द नेक्स्ट सेगमेंट वी विल टॉक about nutrition in human being. So many questions are waiting for board examination from the same topic, so don't forget to subscribe my channel, to like my videos. Thank you, have a nice day.